Hi, welcome to my video on cells and energy. And I'm going to be doing chapter 5, but a little bit out of order. I'm going to start at section 6, and then I'm going to skip back to starting from section 1 on to about section 5. So if you're following along in your book, this is the order I'm going in from chapter 5. So this chapter is called Cells and Energy, and I want to start out with this idea of energy first. In cells, the energy molecule is called ATP. Now, that is an abbreviation. ATP is an abbreviation for this long word, adenosine triphosphate. And ATP, the way it's usually represented in a picture is you have a large adenosine molecule, which is a modified nucleotide. So when you were reading about DNA is made up of adenine, guanine, cytosine, thymine, adenosine comes from a modification of adenine. So you've got this big adenosine molecule, and then you've got 3 phosphates in a row attached to it. These lines right here represent chemical bonds. So here's a chemical bond here, a bond there. There's a third bond, but that third bond is usually drawn as a little squiggly line. That's because this bond is often broken and then it's often rebuilt. When you break that bond, it actually releases energy. And then when you, E-E-N-E-R-G-Y, and then when you rebuild that bond, then you store energy. And so there's something called the ATP Adp cycle. And it looks like this. When you have an ATP molecule and you break it down, you form adenosine diphosphate plus what's called an inorganic phosphate. In other words, we separate this little bond right here, and what you have left is the ADP, and then that little phosphate by itself. Because the bond is broken, you release energy. If you want to reform the bond, then you take that energy and put it back in between those guys. So phosphate plus ADP plus energy. You put those together and that makes ATP. And so this cycle just kind of goes back and forth whenever you need to release energy or storing energy. So breaking the chemical bond releases energy, reforming the chemical bond stores energy. I'll put that in the notes, releases energy. So if you want to know what ADP looks like, ADP is just adenosine. Oh, I don't know if I'll have room there. And then 1 phosphate and 2 phosphates. And then you've got that little extra phosphate. So this is ADP, adenosine diphosphate. So when you stick those two back together, you need energy to create that chemical bond. There's a diagram in your book, and I'm going to look at that diagram real quick. Here it is from your book. This is the Adp Atp cycle, and here's the ATP molecule. And whenever we break the bond, we generate ADP, and one of those little phosphates by itself, energy is released. If you want to stick these two back together, you have to have an input of energy to create ATP back. So this is how basically we use energy and how we build energy molecules. When we need to get energy out of ATP, we break the bond and we use that energy to do whatever cell work is necessary. It could be to have your muscle cells move, to make your heart beat, for you to take a breath, for you to blink your eyes, or for your nerve cells to send a message. Anything that would require ATP or would require energy, forces us to break the chemical bond. Whenever you build molecules and you create chemical bonds, then you store the energy back and you build ATP for use later. So you'll see some questions on the quiz for this chapter talking about the ATP Adp cycle. The big takeaway from this is that when you break down ATP, you get energy. When you put energy back in, there you go. When you put energy back in, you rebuild that ATP molecule. So if I'm going to show you my diagram, well, let's see. I was supposedly getting better at this. Okay, let me start over here. Okay. Let me go back to sharing my screen. Here we go. So this is what you'll see questions on as part of this chapter is this idea of when you break down ATP, what do you get? Adenosine diphosphate instead of triphosphate. You get a free or something that's not linked together, inorganic phosphate, and energy. When you put those three back together, you link that free phosphate back to make ATP. All right, so now that we've talked a little bit about energy, or at least ATP, what about energy in the universe? Well, a lot of cell processes require ATP. As we learned in another video, the exception to this rule are things like diffusion and osmosis. Those don't require any ATP. They do have energy from the universe, but it's not energy as provided for the cell, from the cell. The cell doesn't make diffusion happen, doesn't make osmosis happen. It's a natural process. But there are other things that do require ATP. The cell has to make them happen. And so forming ATP is really important. And so in a future video, we're going to talk about specifically how we form ATP. The rest of this video is going to be dedicated to proteins that kind of guide the process of making ATP. So special proteins that help regulate chemical reactions are called enzymes. They're responsible really for two things, is they speed up chemical reactions, and they also coordinate reactions. Oh, I didn't finish my thought here, chemical reactions. So they speed them up and they coordinate them. So remember that metabolism is all of the chemical reactions that happen inside of a living thing. There are reactions that build. Those are anabolic reactions. And then there are reactions that digest or break things down. Those are catabolic reactions. So many times you have to break something down in order to recycle the parts and build them later. This example is what we call coupled reactions. Coupled reactions means that the beginning of one is dependent on the ending of another, or that they occur at the same time. So a lot of reactions are coupled. A good example would be like when we want to break something down. So for example, if we want to break down glucose. Glucose is a 6-carbon sugar. That means we've got 6 carbons forming the backbone. Ideally, we want to break the glucose molecule into 2 halves, but that doesn't happen on its own. the cell has to invest energy in the form of ATP to make that happen. Well, what we're actually doing is you break down ATP in order to release energy, and the energy from the breakdown of ATP is used to break this bond. So that's a good example of a coupled reaction. This reaction happens first, and it creates the energy we need to make the next reaction happen. Now, in order for this to actually occur at the right speed and at the right time, we have to have enzymes that basically regulate the reaction. So the rest of this video is going to be talking more about enzymes and how they make reactions happen fast enough and how they make them happen at the right time. So enzymes themselves, these are special protein molecules. They are specialized protein molecules that, again, they speed up and they coordinate reactions. There are certain enzymes that are building enzymes, so you can have anabolic enzymes. And then there are other enzymes that break things down, so we can call them catabolic enzymes. Another word for catabolic enzymes is digestive enzymes. This is a more common word than catabolic. So let's talk about some features of enzymes. Some features. Well, when you look at an actual enzyme, they have a special pocket, or you can call it a groove, or a little area where the chemical reaction takes place. And so whatever chemical reaction that enzyme is going to regulate, it occurs in this pocket or this groove. The name of that pocket or that groove is called the active site. Again, that's a quiz question. The active site is the name of the area where the enzyme occurs. So let's say I'm going to draw, this is just some enzyme, and I'm going to draw them like a little Pac-Man. Those of you that are children from the 80s, you know who Pac-Man is. So if this is my enzyme, this right here is the place where the enzyme or the chemical reaction occurs. That's the active site. Every enzyme is specific for a particular substrate. And a substrate is whatever the enzyme works on. So let's say this high wedge here is what my enzyme is actually going to work on. So this is our substrate. So what's going to happen is that my enzyme and my substrate are going to come together. So the substrate fits into the active site. Then the chemical reaction occurs. I get my enzyme back, but now my substrate might have been cut in half. Once the substrate is changed, now we call it the end product. And in this case, I have two of them. So a substrate is converted to one or more end products. The enzyme, and this is another feature, the enzyme remains unchanged, or we can say it is recycled. Now this enzyme can go back and find another substrate and do its action again. Every time an enzyme does its action, whatever its action is, we say that that enzyme has done one turnover. So a turnover is one time the enzyme does its action. So this diagram right here would represent one turnover. If this enzyme went back, found another substrate, and broke it down, that would be 2 turnovers. A third time, 3 turnovers. You get the idea. Most enzymes work incredibly fast. How fast? Well, they can break down up to thousands of enzymes per second. Now, not a single enzyme, but all of them working together, they can break down up to 1,000 per second. So that's like a huge turnover number for enzymes to work. So enzymes have an active site. That active site is where the substrate fits. The substrate is broken down into end products. This thing right here, this temporary structure where the enzyme and the substrate are together, that's called the enzyme substrate complex. Right, that's called the enzyme substrate complex. This is a temporary structure. It doesn't last for very long, but it's an important structure. Something that you should know about that structure is that the substrate, whatever your enzyme's working on, does not fit perfectly into the active site. It's not a perfect fit. The active site is just a little bit too small, just a little bit too small, so that the substrate has to be forced into the active site. Because you're forcing the enzyme into this little space, what that does is it creates stress on the chemical bonds that make up the substrate. Creating stress makes it easier for those chemical bonds to break. So another feature of enzymes is that the substrate doesn't fit perfectly into the active site. The substrate has to force itself, or has to be forced actually, into the active site. And that process puts stress on those chemical bonds, makes them easier to break. Another feature of enzymes is they are incredibly sensitive. And this is probably the biggest, most important feature of enzymes, is that they are incredibly sensitive. Their turnover, okay, remember the number of times they do an action, their turnover rate, it can be impacted by lots of things. And we'll only specify about 3 of them in this video, but they can be impacted by temperature, by the pH of the environment, and by the presence of other molecules. One of your labs, I can't remember if it investigates turnover or turnover in temperature or turnover in pH, but one of your virtual labs definitely looks at how enzymes are affected by one of these. So let me give you some examples of enzymes, because I'm talking in generalities. Well, what about some actual enzymes? Well, here's some examples. Sucrase, that's a sugar molecule that's made up of 2 building blocks. Sucrose can be broken down into glucose and fructose. And the way that we generate these two is that we have to break that chemical bond between them. Now, if we wait long enough, and I mean like a really long time, that chemical bond might break down on its own. But we need to break that bond in order to get the energy that's stored in that bond. So what Mother Nature did was she created an enzyme called Sucrase, which breaks that bond down faster. So if I go back to my Pepsin enzyme, this would be Sucrase. This little pie wedge right here would be sucrose. That's the substrate. This is the enzyme. And then when I'm done, I get my enzyme back. plus I get two little pieces. This one could be glucose and this one could be fructose. So sucrase is an example of a digestive enzyme. Another digestive enzyme, remember that starch is many, many glucose molecules. And so the enzyme which breaks down starch is called amylase. You're probably noticing a trend in enzyme names. Most enzymes end in ase. There's a few exceptions to that rule, and they are the enzymes that we've known about a long time before we came up with the rule of it has to end in ase. An example of something that's the exception to the rule, exception to the ACE rule, is pepsin. We've known about pepsin for a really long time. This is an enzyme which breaks down proteins. So it's still an enzyme, it's just that it got its name before we came up with the ACE rule. So are there any enzymes that build? And the answer is yes. There are some enzymes that build. If you remember earlier, I was talking about the Adp Atp cycle. Well, in order to build ATP, there's an enzyme which takes the available energy and sticks these two together. The enzyme which does the process of building ATP is called ATP synthase. That's an example of not a digestive enzyme, but this is a building enzyme, an anabolic enzyme. So let me talk about how the turnover rate is impacted by temperature and pH. So I'm going to show you a little diagram from your book. All right, here's this diagram right here. Oh my goodness. We're live, folks, meaning I can't run my PowerPoint. There we go. So here's a diagram from your book that talks about how enzymes are sensitive to their environment, specifically temperature and pH. We're going to come back to this diagram, but have it handy. So I think I have to stop sharing and then reshare my screen to make this work. There we go. So we'll talk about temperature first. So every enzyme has an optimum pH. I'm sorry, an optimum temperature. And that's the temperature where you have the best turnover number. So let's make a graph here. And let's say this is turnover number or turnover rate. That's the same thing. Turnover number is the same thing as turnover rate. And let's just say this is in the thousands per minute. So this would be 1,000 per minute, 2,000, 3, 4, 5,000 per minute. And then this down here, this is temperature in degrees Celsius. So this is 10, this is 20, 30, 40, and 50. So if we graph how a typical enzyme works, we might see a graph something like this. So that anywhere that we're above zero, zero's right here, anywhere you're above zero, you have some activity. So our enzyme has a range where it works of about 10 to 50 degrees Celsius, but it doesn't always work at the same turnover level between 10 and 50 degrees. Right here, the top of this graph, which if I was to draw this line down, that's about 37 degrees Celsius. The top of this graph is where I have the best turnover number, about 3,000 per minute, whatever job this enzyme does. That would be an example of the optimum temperature for this enzyme. If we look at this part of the graph, the enzyme is not working as well, but as the temperature increases, it's starting to work better and better. So the general trend is if you increase the temperature, you increase the turnover. So enzymes have a tendency to work better and faster as the temperature increases. The reason is an increase in temperature is adding energy. So literally all molecules, including your enzymes, start to work faster as the temperature increases. The enzyme turnover number increases all the way until we get to 37 degrees, which is our optimum temp for this enzyme. And then after that, even though it gets hotter, our enzyme isn't working as well. Now, what is interesting is that right here, this little dot at 30 and this little dot at 40 degrees temperature, they're working at the same level. However, here we're getting better, but on this side, we're getting worse. And as we continue getting hotter and hotter, notice that the enzyme activity starts to go down. After the optimum temperature, the enzyme begins to experience physical damage. So after the optimum temperature, then what happens is that the active site, remember that's the part of the enzyme that actually has where the chemical reaction occurs, the active site begins to melt. It loses its shape. And so the fancy word for that is we say that the enzyme begins to denature. This is a word that you're going to see on your quizzes for this chapter. What does it mean to denature an enzyme? That means that the active site has changed its shape. When the active site melts or changes shape, you lose the ability to do your job. So your turnover number starts to decrease. When an enzyme is denatured, this is a permanent, irreversible condition. So in other words, we can't heat this environment up to 40 and then cool it down and have our enzymes start working. Think about when you cook an egg. Egg white is all protein. And so you can't uncook an egg. You can't uncook, in a sense, an enzyme. You can have an enzyme that is partially denatured, like right here. So it's okay to have an enzyme that has partial function. Or if you get hot enough, your enzyme loses all activity. So like right here on the graph, it's 0. So our enzyme is completely melted. It's fully denatured. So looking back at our graph from before, here's our graph from before. Looking at the top graph, this shows enzymes from 2 different sources. So here's the rate of the reaction. That's the same thing as the turnover number. And here's the temperature scale on the bottom. The optimum temperature for this particular enzyme is somewhere around 37 degrees Celsius. That probably doesn't surprise you if you know that 37 degrees Celsius happens to be body temperature. So the enzymes inside of humans, they seem to work best around 37 degrees Celsius. Once you get higher than 37 degrees Celsius, look how sharply the function falls down. So as soon as we go past the top of this graph, our enzymes denature. This is why high fevers for a prolonged period of time can be so dangerous, especially for the hottest part of your body, which is your brain, because it is literally the most metabolically active organ. in the body. So if your brain gets too hot with a high fever, you can actually cause brain damage to some extent by cooking the enzymes in your brain. The nice thing is that we have some ability to clean up denatured proteins, but if you stay too high for too long, you can cause permanent brain damage, even death from a really high temperature. Compare that to enzymes from bacteria that live in hot springs. Their optimum temperature is way above what we could tolerate. So hot temperatures don't kill these bacteria. In fact, they're very comfortable, even going up to like 70, 75 degrees, which is pretty hot. It's not quite boiling, but it's considered to be a pretty warm temperature for most other living things. Other living things, they don't tolerate high temperatures as well as these particular bacteria. Let's talk about pH, and we'll come back to this graph again. I think I'm getting a hang of this camera, you guys. So let's talk about pH. So enzymes are much more sensitive to pH than what they are temperature. There is an optimum pH for every enzyme, and that's, of course, where the enzyme has the highest turnover. Okay, has a high turnover rate. But what we're going to find out is that enzymes, they can denature if the pH is too far above or too far below the optimum. So I'm going to put a pH scale here. And you've probably heard of the pH scale before. It measures how acidic something is, and it's based on a scale of 1 to 14, 7 being right in the middle. Anything above 7 is considered a base, and anything below 7 is considered an acid. And the further you are from 7, the stronger you are. So strong acids are on this end, strong bases are on this end. The closer you are to 7, the weaker you are. If you have a pH of exactly 7, you're not an acid, you're not a base, you're perfectly neutral. So let's consider an acid or an enzyme that would work, let's say, in your stomach. Your stomach has a pH of around 2. So if I look at a particular enzyme, and of course this is turnover. Okay, so it gets better as you go up. If I look at an enzyme that works in the stomach, I would expect it to have an optimum pH of around 2, because your stomach has got lots of hydrochloric acid, and so hydrochloric acid has a pH of about 2. So it makes sense that an enzyme that works in your stomach would work well at a pH of 2. And by the way, a pH of 2 is really, really strong. But if we have a separate enzyme, let's say that works in your intestines has a pH much higher than the pH of your stomach. And so the optimum pH of a separate enzyme might be closer to around 8.5. So this would be something that works in your stomach. And this would be more like something that works in, oops, I can't spell intestines, or an intestinal enzyme. So as I mentioned, these guys denature if you go too far above or too far below pH. So the active range for an enzyme that works in your stomach is going to be limited to around 2. If you get too far above or below 2, your enzyme will be denatured. It can be natured partially or fully. So unlike the temperature graph, you can never get better once you go on either side of the optimum range. The same is true for the enzyme that works in your intestines. That's where it works best, somewhere between 8.5 and 9. If you get too far above or too far below, your enzyme begins to melt, but it doesn't melt due to heat. It melts due to an extreme change in pH. All right, I am at my time limit for this video. So I'm going to stop here talking, finishing up my discussion about enzymes and temperature. I'm going to go back to that chart real quick because you'll see this on the exam or the quiz associated with this chapter. Here we go. So the top graph kind of shows what we were just drawing. This is the enzyme pepsin, the one that works in your stomach. And this is the enzyme trypsin, which works in your intestines. And so the way that we control digestion is by changing the pH of the environment. Pepsin digests proteins in the stomach, so its pH range where it goes to work is going to be a lot lower. Once food leaves the stomach, it's going to enter the intestines. Now, the stomach has a lining that is made to have a very low pH, but the intestines, their lining is much thinner. So the gastric juice that comes out of the stomach has to be neutralized so that you literally don't burn a hole in your intestines. Also, you don't want to digest your intestines with this active pepsin. So in your intestines, you get a little bit of baking soda, believe it or not, a little bit of bicarbonate that's added to your intestines coming from your pancreas, the same place that you get insulin and glucagon. The pancreas sends a little bit of baking soda, which changes the pH of the environment. That changed of pH turns off pepsin, but we still need to digest protein. So now we have added a separate enzyme to continue the digestion of protein in the intestines, but this particular enzyme works better at higher pH. So it's an example of how we use the environment to control enzyme activity. All right, that's it for this video. Remember, I'll post my notes so that if you want to follow along with the notes with the video, you can do that. Thank you guys for watching.